Okay. Thank you, to Angus for the invite to come across and speak to you guys, and thanks to Marcus for that great introduction to us start thinking about soils and carbon. I'm a soil scientist, so I love soils. I think it's probably your most valuable resource you have in your orchards. And in case no one else knows how important soils are, the UN has declared this year, 2015, as the International Year of Soils. So around the world, we're celebrating how important they are to us. Okay. Carbon is critical for our soil structure and function. It enhances a multitude of services that our soils provide to us, including water storage, nutrient mineralization, and it fuels our soil microbial communities. So within the PIPS soil carbon program, we were aiming to better understand the links between soil carbon, soil health, and soil functions in order to, in future, perhaps we can optimize these functions for orchard production. So in the program, we conducted a survey of Australian apple orchard soils to establish the status of current soil carbon stocks, to see if we can determine any changes in soil carbon with time, and to establish the relationships between soil carbon and soil functions. And to do this, we collected soils from all of the major apple orcharding regions in Australia, as depicted on the survey map. And at each of these locations, we selected one orchard block for sampling for a total of 10 sites across the country. And so first, when we conducted the survey, we had to establish a sampling protocol which would account for all the spatial variability that exists within an orchard. And we sampled in multiple depth increments down to a meter depth so we could calculate really deep stocks of carbon. So we collected soils from the orchards, took them all back to New Zealand where we could analyze them in our labs. And we performed a suite of measurements looking at soil carbon, soil nitrogen, microbial activity, and diversity. And I'll just note here that we measured both total carbon concentrations and a measure of labile carbon, which with our methods was, you can measure it several different ways, but we used a hot water extraction method, which was developed as an indicator of soil quality in New Zealand pasture soils. So soil carbon stocks showed wide variation across all the orchard blocks. And in this figure, the sites are listed roughly west to east across Australia, with Tasmania stuck on the end. And so total soil carbon stocks to 1 meter depth range from 60 to 260 tons of carbon per hectare in these orchards. And you'll note the really high carbon stocks, Stanthorpe, Gippsland, Donnybrook, and Management, those soils also had really high carbon to nitrogen ratios, and they also had visible pieces of char in those soil samples. So these really high carbon stocks are likely to be attributed to the presence of charcoal in these orchard blocks. And this charcoal may originate from the historical land clearing or maybe from orchard redevelopment practices. But I just thought I'd point that out because almost everybody objects to Stanthorpe having the highest soil carbon stocks of all the orchard regions, but we think that there actually just was a lot of charcoal present at this site. So our variety of soil health parameters also varied widely amongst all the orchard blocks that we measured. But we did, when we were trying to see how they're related to each other, we did see some strong positive relationships between them. And this is a table showing correlation coefficients amongst the variables. So the closer the number is to 1, the stronger the two variables are related to one another. So our measures of labile carbon, total nitrogen, potential nitrogen mineralization, and microbial activity were all strongly correlated with one another. And these variables all serve as indicators of soil carbon regulation and nutrient supply in our orchards. What's particularly interesting to note is that our measure of labile carbon was much more strongly correlated with microbial activity and potential nitrogen mineralization than were our measures of just total carbon. And so labile carbon represents just a very small fraction of total carbon in our soils. And this figure is showing concentrations just for the top 0 to 10 centimeters in our orchards of total carbon and labile carbon. We have our total carbon scale on the left-hand y-axis, and this is representing the solid green bars, and this scale is going from 0 to 50. And on the right-hand axis, we have our measures of labile carbon, which are the sort of open blue bars. And you can see there's a 50-fold difference in scale. So the labile fraction is a very small fraction of carbon. And what's also obvious is that the variation in our labile carbon in no way matches the variation that we're seeing in our total carbon. So again, for example, if we take the Stanthorpe orchard, which was our high carbon stocks. That also has quite high total carbon in the orchard, but we can see has one of the lowest concentrations of labile carbon. So what this is showing is that a large proportion of the carbon in the Stanthorpe orchard is recalcitrant, and it's just not available to soil microorganisms. But because our measure of labile carbon was much more strongly correlated with microbial activity and potential nitrogen mineralization rates, it's proving to be a more sensitive indicator of potential nutrient supply in our soils than is just total measures of total carbon. And because it's actually quite simple to measure in the lab, much more so than potential nitrogen mineralization rates or microbial activity, it might be a useful proxy to include in soil testing for these measurements. Our DNA analysis of the soil microbial community composition revealed that the orchards differed in their microbial community composition, and that's depicted in these figures. This is the result of a multivariate statistical analysis of the relative gene abundance for different microbial groups and different genes involved in nitrogen cycling. And so the figure on the left is showing the separation of the orchard sites. So in this figure, the further apart they are, the more different their microbial community composition is. And on the right are the vectors underlying these separations. So it's difficult to interpret, even for me, but for example, moving up in an upwards direction, it's predominantly due to the presence of a nitrate reductase gene, which is a gene involved in reducing soil nitrate to nitrite. However, what was I going to say? This is showing that the communities differ, and we tried to link these differences in the gene abundances we measured with the soil health properties that we measured across all the orchards, and no correlations were apparent. So we couldn't establish relationships between what was there to the properties that we measured. So it could be things like soil type, environmental conditions, or the seasonality of sampling, much more important drivers for our microbial community composition. And so all researchers will say that you need to do more work, but that's definitely true in this case. We can, at this point, link our microbial community composition to the functions that are happening in our soil. So what we really need are further work with more detailed comparisons and control conditions before we can start linking what microbes are present to the functions we're getting from our soils. And so lastly, in the project, we're aiming to see if we detect any changes in total soil carbon stocks over time. So we did some intensive sampling in 2 orchard blocks in Shepparton, Victoria, and 1 block in Huonville, Tasmania. And when the PIPS program began, we sampled at the end of 2009, and then as the project was wrapping up, we returned to these orchard blocks to resample at the end of 2013. And from our paired samplings 4 years apart, we did not detect any net change in soil carbon stocks down to a meter depth. And so even this maintenance of carbon stocks under orchard production is a positive outcome, because at least we can see soil carbon does not appear to be declining under orchard production, as is quite common for many other agricultural practices. However, we should caution that 4 years is not a very long period of time to detect what might be very small changes against large background stocks. And so we would really advocate resampling over long time intervals and perhaps continued sampling over additional deep soil carbon profiles to verify whether any changes are occurring in carbon stocks under orchard production. So in conclusion, our work in the PIPS soil carbon program has established baseline measures of soil carbon stocks and quantified soil health metrics across across the Australian apple orcharding regions. Our database shows the extent of variation that exists in these properties in apple orchards. One of our most significant findings was that our measure of labile carbon was quite a sensitive indicator of potential nutrient supply, much more so than just total carbon. And lastly, we would always advocate that it's important to maintain or enhance soil carbon stocks so that we can sustain these important soil functions. And lastly, special thanks to all the growers who let us come onto their orchards and take away some of their soil. And thanks to our funders.